Hello dear students, how are you all? I hope you people are all healthy and fine. As we know, India has been locked down till 14th of this month, but it is my sincere request to all of you is please don't lock down your learning process. Today in this video clip we are going to understand the very important concept of second chapter, that is structure of pollen grains. Why it is so important? Because this question was repeatedly asked in the previous year question paper in the board examination. In 2014, it has been asked for 3 marks. In 2016, it has been asked again for 3 marks. And in 2017, it has been asked for 5 marks. That makes this concept so important to understand. And there are so many questions appear from this particular concept in the competitive exams like NEET, AIMS and the JIPMER. Now let us understand the structure of the pollen grain. Before we discuss the detailed ultrastructure of a mature pollen grain, we have to understand what is this pollen grain. Pollen grain is a male gametophyte, and this male gametophyte is getting synthesized, getting nourished and getting matured inside the pollen sac. Now the question arises where this pollen sac is situated. This pollen sac is situated inside the anther, and anther is a part of androecium. So these pollen grains are getting nourished, synthesized inside the anther. We know anther is bilobed, dithecous. It consisting of 4 layers of walls. Within that 4 layers of walls, there is a pollen sac. Let us recall what are the 4 antheridial walls. The outermost layer is epidermis. Then we have got endothecium. After that we have got middle layer. Then lastly, to the inner side of the anther, we have got the tapetal layer or tapetum layer. Within that tapetum layer, there is a pouch-like structure. There is a sac-like structure. That sac-like structure is nothing but the microsporangium, or it is simply called as pollen sac. Inside that pollen sac, the pollen grains are getting synthesized and getting matured. Now, the detailed study of pollen grains, it is called as palynology. Palynology. Palynology, it is a branch of botany that deals with the study of the pollen grains. Now, as we have discussed earlier, pollen grains are the male gametophytes. They are not the male gametes. They are the male gametophytes. That means within the pollen grains, the male gametes are getting synthesized. Pollen grains, they are actually carrying the male gametes. That is why they are considered as male gametophyte. And the detailed study of pollen grains, it is considered as palynology. And the person who is expert in the studies of pollen grains, he is called as palynologist. 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 The person who is expert in the study of pollen grains. Now, let us understand. Pollen grains, they are of various shape, various size in different plants. But the general or widely accepted diagram of the pollen grain is of spherical shape. This spherical shaped pollen grain, it is covered with 2 protective layers. The outer protective layer, it is called as exine, and the inner protective layer, it is called as intine. That means the outer layer of pollen grain, it is called as exine, and the inner layer of the pollen grain, it is called as intine. Intine is completely different from the exine, but both are protective in their function. Structurally, they are different. Chemically, they are different, but functionally, they are one and the same. Both are giving protection to the pollen grain. Now, this intine, it is made up of 2 substances. One is cellulose and pectin. Cellulose and pectin, that makes the inner layer of the pollen grain. Whereas exine is made up of a very special substance. That special substance is nothing but Sporopollenin. Sporopollenin. We'll understand what is that sporopollenin separately here. Now, intine is made up of cellulose and pectin. Exine is made up of sporopollenin. Now, if you carefully look both the exines as well as the intine, exine is non-uniformly distributed. Here and there we have certain gaps. It is not completely uniform layer of a pollen grain. It is non-uniform layer. It is non-uniform. Whereas intine, it is completely uniform. We don't have any gaps in the intine. It is uniform. Uniform. But both are protective in their function. Now, exine, as we have discussed earlier, it is made up of a particular substance, that is sporopollenin. Let us understand what is this sporopollenin is. This sporopollenin is considered as a highly resistant material. It is the highly resistant material. Sporopollen is actually a protein that getting deposited on the outer side of the exine. That means exine is deposited with the sporopollenin, and this sporopollen is a highly resistant material, very strong material, very tough material, and this sporopollen cannot be degraded. Cannot be degraded by acids, bases, enzymes, even UV radiation. Even UV radiation cannot degrade it. Ultraviolet radiations, they cannot damage the sporopollen. That is why most of the pollen fossils, most of the ancient pollen grains, they are still preserved under the soil due to the presence of this sporopollen. Sporopollen helps in pollen fossilization. Pollen fossilization. Now, this sporopollen, it is also not uniformly distributed. Here and there we have got certain openings. These openings are called as germ pores. They are called as germ pores. Now, these germ pores' main job is the pollen tube after fertilization. When this pollen grain is dropped on the stigma, then the pollen grains start getting germinated. That process is known as pollen tube growth or pollen tube germination. Now, during that pollen tube germination, this tube comes out from this germ pore. This may be the pollen tube. That is the future process of this pollen grain. This is a pollen tube. We need not label this in this diagram. Just for our understanding, I made it here. Now, germ pores are helpful for the growth of pollen tube. Then the question arises, why it is necessary to have a germ pore? Because all over the outer surface of the pollen grain, sporopollenin is deposited. The pollen tube cannot grow from here because there is a sporopollen here. Sporopollen is a highly resistant material that will not allow the growth of the pollen tube. That is why here we don't have the deposition of the sporopollen. Through this route, the pollen tube can grow in future. Now, pollen grains may be classified into various categories based on the number of the presence of the germ pores. If there is only one germ pore in the pollen grain, then it is called as monocarpate. If two germ pores are there, then it is called as bicarpate. If there are three germ pores in the pollen grain, then they are called as a tricarpate. Now look at the diagram. How many germ pores are there in this pollen grain? There are three germ pores. This is first, second, and the third. There are three germ pores. That means this pollen grain is a tricarpate pollen grain. Based on the number of the germ pores, pollen grains can be classified into various categories like monocarpate, bicarpate, tricarpate, tetracarpate, pentacarpate, hexacarpate. Like that we can classify the pollen grains. Now coming to the internal structure of the pollen grain. Pollen grain is protected with the two layers. Outer layer is exine, inner layer is intine. One more very important point that we have skipped here. Intine is semi-permeable. Semi-permeable. Whereas exine is non-permeable. Intine is semi-permeable and exine is non-permeable. That means intine is behaving just like the plasma membrane of our cell. Now, let us study the internal structure of this pollen grain. Pollen grain is always a haploid structure. Pollen grain is a haploid structure. In this haploid male gametophyte, we have two cells located. The upper cell, the swollen cell, the large cell, the bulgy cell. This cell is called as vegetative cell. It is also called as tube cell. It is also called as tube cell. And the name itself giving the meaning, vegetative cell, tube cell, that means here plenty of food is stored. This is food, plenty of food is stored here. That is why it is called as vegetative cell. And in future, after pollination, this cell will germinate into a pollen tube. That is why it is called as tube cell. Vegetative cell's main job is to nourish. Why it is nourishing cell? Because it is having abundant resources of food in it. That is why it is a nourishing cell, to nourish the generative cell. Generative cell. The main job of the vegetative cell is to nourish the generative cell. This vegetative cell, it is also called as tube cell, because in future after pollination this will develop into a pollen tube. That is why it is called as tube cell. Here plenty of food is reserved, plenty of food is stored here, abundant amount of food is stored here. That is why it is called as vegetative cell. Now coming to the lower side here. Here we have one more spindle shaped small cell as compared to the vegetative cell. This cell is called as a generative cell. This generative cell, it is also called as a gamete cell. Gamete cell. The name itself again giving the meaning here. Gamete cell results into the formation of gametes. This synthesizes or it produces, generative cell produces male gametes. It produces the male gametes. Vegetative cell main job is to nourish the generative cell, and generative cell's main job is to produce the male gametes. It is at the lower side, spindle shape. Now to separate the vegetative cell from the generative cell, there is a thin layer of wall. It is a bilayered wall because there are two layers of wall. A thin layer of wall. This layer of wall, it is called as callase wall, which is made up of a substance callose. Callose makes the callase wall that separates the vegetative cell from the generative cell. Now let us understand the diameter of this pollen grain. This pollen grain, it may be 50 to 70 microns in the diameter. 50 to 70 microns in the diameter. Please remember here the most important point is sporopollenin. Sporopollenin, this question is asked in the 2018 NEET question paper. NEET question paper, and the question was, what is the significance of sporopollenin to the pollen grain? That question was asked. I hope this video will help you to revise the things that you have studied in the classroom. Now, in our next video, we'll understand one more very important concept. Till then, stay at home, stay safe. Thank you.